is our general, the open area. And then we have our chromatography room, this one. Chromatography room. We have the HPLC. This one we are using it so much in analysis of aflatoxins. You know, we are having some challenges with aflatoxin, but this is the guy we use to handle aflatoxin analysis. We do HMF in honey. We also have the GC. This one we do methanol with it. Remember recently we do methanol analysis in the alcoholic beverages. This is the one we use for. And now this is our balance. We do our weighings from this. Yes. And then from that we have the general analysis room. It's the general analysis area, where we do these other titrations and the classical methods we do in this generalist area. The moisture in the different foods, we do this. We use this is an oven for moisture determination. We have furnaces. We have 4 furnaces for ash and then acid insoluble ash in foods, things like that. The ovens, you have these are 2 and then these other ones. That one and the other one. And actually right now I'm extracting for aflatoxin analysis. We have maize grains and then some other floods you'll see on the other side there. Currently I'm extracting this for aflatoxin analysis. Yes, we have the different equipment, centrifuge for that, whatever I'm going to use, centrifuge and all that. And then we have other equipment. For example, this one is for protein analysis, this one. The other one is for fiber. Then the fridges where we keep our standards and all that. Control samples. This one. In oil there is water. You don't believe that in oil there is water. So when we are doing water content, or you see after processing, they wash the. You see after oils have a lot of acids and all that during processing. So they put the sodium hydroxide, try to neutralize it, and then wash out that sodium hydroxide. So they use water to wash. But now it's supposed to contain very minimum amounts that are acceptable, because when it's too much then the oil will go bad very fast, it will oxidize. So this equipment we use it for analysis of moisture content in oil. It's a vacuum oven. This one, yes. Then you can come to now microbiology. You can hand over to.
I'm James Chivumbi and I'm the in charge for the microbiology lab. Yes. So our process starts from this side. So you have issues with space, and we try to manage. So the process starts from our weighing room. Maybe you can come and have a look. Yes, this is where we do the weighing from for everything that we use. Yes, especially what we call culture media. Culture media is, in simple terms, is food for microorganisms. Yes. So after weighing from there, we have the autoclave where we do sterilization. We sterilize it from here, and then this is our fridge for the storage of the media which has been prepared. So after weighing, we also weigh the samples that we are going to work on from here. So the sample preparation happens from here, and then from here we go to the other side where we do the actual analysis. Yes. We have our small room, we call it the washroom, where we do the washing of glassware and everything that we use. Yes. Then we have a small stove here for finished samples, where we put samples after analysis. Yes, there's a small space here. That's what we use. So this is like our main lab, this side. So in here we have the biosafety cabinet. So the analysis of the samples happens from here. It is a sterile environment. So this is which is where we do the actual analysis from. Then after preparing our analysis, we put in an incubator. Now incubators, we have many because we incubate at different temperatures. So we have this, and those two that you saw in the corridor there, those big ones. So we incubate at different temperatures for a particular time. Then from there, after incubation, we have this one here. It is called an automatic colony counter. It is this one here. Yes. It is where we read our results from. When you get our results, we put in the system, and then we do the certificates and everything else that goes on.
So all the colonies of viruses, bacteria.
Yeah. Anyway, we don't do viruses, but bacteria. Yes.
Okay.
Yeah. You can see these are samples which are going to be worked on.
Okay.
We still have issues with the space, but we are trying to handle for now. Yeah. So these are samples that have come from clients which are supposed to be worked on. Yeah. Mainly we handle foods, because those are the most common samples we receive here. Yeah, they are mainly food, but we also have other samples, but it's mainly food, especially maize flour, honey. We have fermented beverages, wine. Yeah, then you have animal feed. Yeah, this region usually has most of such samples. So that's what we usually mostly handle from here. Yeah. Unless you have any other question. But in simple terms, that's what we do.